It feels like every day we're getting closer and closer to this sci-fi future we've always imagined, damaged brains and spinal cords being repaired.
Yeah.
And it's starting to feel less like science fiction and more like, well, science.
Yeah, it really is remarkable. For a long time, the idea of regenerating neurons seemed almost impossible. But this research from MIT, it is a major leap forward.
Absolutely. So this deep dive is all about understanding just how big of a leap this is. We're looking at a paper published just days ago on March 13th. And honestly, it has me a little giddy, turning skin cells directly into neurons, bypassing the usual stem cell rigmarole. It's kind of mind-blowing.
Yeah, what's really fascinating here is that this isn't just about speed and efficiency. That those are huge factors.
Right.
This direct conversion method, it avoids a lot of the roadblocks that have really plagued regenerative medicine for years.
Hey, so walk me through this. What were those roadblocks and why is this new approach so different?
Well, for a long time, the standard approach to generating neurons in the lab, it involved creating what are called induced pluripotent stem cells, or iPSCs.
Yeah.
These are essentially adult cells, like skin cells, that have been reprogrammed back into a stem cell-like state.
So kind of like rewinding the developmental clock of the cell.
Exactly. But this process is notoriously finicky, it's time-consuming, the efficiency is often low, and sometimes the cells get stuck and never fully revert to that pluripotent state.
So a lot of trial and error and a lot of wasted time. I can see why scientists would want to find a better way.
Absolutely, and that's really where this new research shines by converting skin cells directly into neurons. They've bypassed the iPSC stage entirely, and the results are astounding, over 100 times greater efficiency.
That's a staggering difference. So how are they doing it? What's the magic trick?
It's surprisingly elegant, actually. They use a modified virus to deliver 3 key transcription factors directly into the skin cells. Think of transcription factors like genetic switches that control which genes are turned on or off, basically dictating what a cell becomes and how it behaves.
So these specific transcription factors, they're essentially rewriting the skin cell's genetic instruction manual, telling it, hey, you're not a skin cell anymore, you're a neuron now.
Precisely, the researchers have pinpointed the 3 essential ingredients for this transformation: NGN2, ISL1, and LHX3.
Just 3 ingredients and you get a neuron. It sounds almost too simple.
Well, there's one more clever trick they use to boost efficiency even further before introducing those transcription factors. They give the skin cells a proliferation boost.
Proliferation boost now, that sounds interesting.
They use 2 additional genes, P53DD and a mutated version of HRAS, to stimulate rapid cell division before the conversion process.
So it's like preparing the ground before planting seeds. You create those optimal conditions for growth and change.
That's a great analogy. The researchers found that these hyperproliferative cells are much more receptive to that reprogramming power of those transcription factors.
It makes sense you're giving the cells a head start on their transformation journey, but does it actually work? Do these reprogrammed cells really behave like neurons?
The results are incredibly promising in mouse studies. These converted neurons were successfully transplanted into the brain, where they integrated with existing brain tissue, formed connections with other neurons, and even exhibited electrical activity.
Wait, so they put them into actual mouse brains and the cells function like regular neurons? That's remarkable.
It really is. While the efficiency in human cells is currently lower, around 10 to 30%, it's still a significant improvement over previous methods.
Okay, that's encouraging. But what about applications in humans? What kind of conditions are we talking about treating here?
Imagine the possibilities: spinal cord injuries, ALS, Parkinson's disease, Alzheimer's. The potential is vast. And what's particularly exciting is the prospect of personalized medicine using a patient's own skin cells to create neurons, eliminating the risk of rejection.
Wow, that's a game changer. But I have to ask, what are the challenges involved in translating these promising results from mice to humans? There must be more to it than just growing human neurons in a dish.
You're right, there are still hurdles to overcome, and the process won't be simple. We'll need to understand how these new neurons integrate into the complex circuitry of the human brain.
It's not just about replacing lost cells. It's about making sure they function correctly within that intricate network, right?
Exactly. That brings us to the next stage of this research journey: understanding the potential and the complexities of applying this technology to real-world medical treatments. Picking up where we left off, one of the big challenges with applying this technology to human therapies is ensuring that these newly created neurons not only survive, but thrive in their new environment.
So it's not enough to just plop them into the brain and hope for the best.
Not quite. The human brain is an incredibly complex and dynamic organ. Simply introducing new neurons doesn't guarantee they'll integrate properly or function as intended.
That makes sense. It's like trying to add a new instrument to an orchestra mid-performance. You need to make sure it's in tune and playing the right notes.
That's a great analogy. We need to ensure these new neurons connect with the right circuits, receive the appropriate signals, and really contribute to the overall harmony of brain activity.
And how do researchers go about figuring that out? It sounds like a delicate and intricate process.
One approach is to start with in vitro studies using human brain organoids, which are essentially tiny 3D models of brain tissue grown in a lab.
So kind of like mini brains in a dish. That's pretty wild.
It is quite remarkable. These organoids allow researchers to study how these new neurons behave in a more realistic environment outside of a simple petri dish.
I imagine that gives them valuable insights into how the cells might integrate into an actual human brain.
Exactly. It helps them understand things like how well the new neurons form connections, how they respond to different stimuli, and whether they exhibit any unexpected or potentially harmful behaviors.
It sounds like there's a lot of testing and fine-tuning involved before we see this technology being used in actual patients.
Absolutely. Safety is paramount, and researchers need to be incredibly thorough in their evaluations.
But let's say they can get these neurons to integrate perfectly and function as intended. Are there any other potential challenges we need to consider?
Well, even if the technical hurdles are overcome, there are still logistical and ethical considerations. For example, the process of generating these neurons is still quite complex and specialized.
So scaling up production to meet the potential demand could be a challenge.
Precisely.
Yeah.
We need to find ways to streamline the process, make it more efficient, and ensure that it can be carried out reliably on a larger scale.
And what about access? We've touched on the idea of personalized medicine using a patient's own skin cells to create neurons, but realistically, how feasible is that on a large scale?
That's another critical question. While personalized medicine offers incredible potential, it also presents logistical and economic challenges.
It makes you wonder if this treatment becomes available, how do we ensure that everyone who could benefit from it has access, regardless of their financial resources or geographic location?
Those are complex issues that will require careful consideration and thoughtful solutions. It's not just about the science, it's about ensuring equitable access and distribution of these potentially life-changing therapies.
It's a reminder that scientific breakthroughs, while exciting, often raise as many questions as they answer.
That's the nature of scientific progress. It's a continuous journey of discovery, refinement, and adaptation.
So what's next for this research? Where do scientists go from here?
Well, one of the key areas of focus will be optimizing the conversion process in human cells. Remember, the current efficiency is around 10 to 30%.
Right, so only a fraction of those skin cells are successfully transforming into neurons. How do they improve that?
It's a multifaceted challenge. Researchers will be looking at everything from fine-tuning the delivery of those transcription factors to identifying specific cell types or conditions that might enhance conversion efficiency.
So it's back to the lab, tinkering with the recipe, so to speak.
Exactly. It's about finding the perfect combination of ingredients and techniques to achieve the highest possible yield of functional neurons.
And once they've cracked that code, what's the next step?
Well, assuming they can achieve a high and reliable conversion rate, the next big hurdle will be testing these neurons in human clinical trials.
Ah, so this is where things move from the lab bench to the bedside.
Exactly. Clinical trials are essential to determine the safety and efficacy of any new medical treatment, especially one as groundbreaking as this.
I imagine these trials will be complex and take a significant amount of time.
Researchers will need to carefully select patients, monitor their progress closely, and meticulously analyze the data to determine whether these new neurons truly offer a viable treatment option.
It's a long road, but the potential rewards are enormous.
They are. And while we await the results of those trials, there's another fascinating aspect of this research that's worth exploring.
Oh, I'm intrigued. Tell me more.
This direct conversion technique isn't limited to just creating neurons. Scientists are also exploring its potential to generate other types of brain cells like astrocytes and oligodendrocytes.
Now, those are names I haven't heard in a while. Remind me what they do again.
Astrocytes are like the support staff of the brain. They help nourish and protect neurons, regulate blood flow, and play a role in immune responses.
So kind of like the unsung heroes of the nervous system.
Exactly. And oligodendrocytes are responsible for producing myelin, the fatty substance that insulates nerve fibers and allows for rapid signal transmission.
Ah, so they're crucial for communication within the brain.
Precisely. And the ability to generate these different types of brain cells opens up a whole new realm of possibilities for treating neurological disorders.
It sounds like this research could have far-reaching implications impacting not just conditions that directly affect neurons, but also those that involve damage or dysfunction in other types of brain cells.
You're absolutely right. This research is opening doors to potential treatments for a wider range of conditions than we initially imagined.
This is truly revolutionary stuff.
It is, and as we delve deeper into this research and its potential applications, it's important to remember that we're not just talking about scientific advancements, but also about profound changes in how we understand and interact with the human brain.
It feels like we're on the verge of a new era in neuroscience and medicine.
I agree, and as with any major paradigm shift, there will be both challenges and opportunities along the way.
And speaking of opportunities, what are some of the specific ways this research could translate into real-world treatments in the future?
That's where things get truly exciting. Let's explore those potential applications in our final segment.
All right, so we've covered the why and the how of this research, but now let's talk about the what if. What does this technology actually mean, for treating these neurological conditions?
Yeah, if we connect this back to the bigger picture,
Yeah.
this research has the potential to revolutionize how we approach a wide range of conditions. We've mentioned some of the big ones, spinal cord injuries,
Yeah.
ALS, Parkinson's, even stroke, and Huntington's disease, but it really goes beyond that.
I'm ready to have my mind blown. Give me some specifics. How could this actually play out in a treatment setting?
Okay, well, let's take spinal cord injury as an example. Currently, there's no way to regenerate those damaged neurons, which leads to that permanent paralysis. This direct conversion technique could offer a way to actually replace those lost neurons, potentially restoring function and mobility.
It's almost hard to wrap your head around, we're talking about potentially reversing paralysis. That would be life-changing for so many people.
Yeah, and it's not just about replacing the neurons. Remember, we talked about astrocytes and oligodendrocytes. They play such a crucial role in supporting and protecting neurons. Being able to generate these cells could be really vital for creating a healthy environment for those new neurons to thrive.
It's like rebuilding the infrastructure of the nervous system. Fascinating.
Exactly. And because we can use a patient's own skin cells, there's no risk of rejection. It's personalized medicine at its finest.
Yeah, that's one of the aspects that makes this research so compelling, but I'm also curious about the timeline. How far off are we from seeing these types of treatments become a reality?
Well, that raises an important question. While the science is incredibly promising, we need to be realistic about the road ahead. It takes time to translate these lab discoveries into safe and effective therapies.
I get it, we don't want to give people false hope, but even if it's years down the road, the potential is still incredibly exciting.
Absolutely. And in the meantime, the research continues to advance at a rapid pace. There's so many avenues to explore, so many questions to answer.
Like what? Give me a glimpse into the future of this research.
Well, one area of intense interest is improving the efficiency of converting human cells into neurons. We're currently at 10 to 30%, which is a good start, but there's definitely room for improvement.
Right, the higher that percentage, the more effective the treatments will be.
Precisely, another exciting area is developing more targeted delivery systems. Right now, they're using a modified virus to deliver those key transcription factors. But researchers are exploring other more precise methods, like nanoparticles or even gene editing techniques.
So it's about refining the tools, making the process more precise and efficient.
And then there's the whole area of bioprinting. Imagine being able to literally print functional neural tissue, creating these custom-designed implants for specific areas of the brain or spinal cord.
Okay, that sounds straight out of science fiction, but is that even remotely possible?
The field of bioprinting is advancing rapidly, and it's certainly within the realm of possibility. It's a truly exciting time to be following this research.
Well, you've definitely given us a lot to think about as a listener. I'm left with this sense of awe at what science is capable of achieving, but also a sense of responsibility. This technology has the potential to really change lives, but it's important that it's developed and used ethically.
Oh, I completely agree. As this technology advances, we need to have open and honest conversations about its implications, ensuring it's used for good and benefits all of humanity.
So for all of our listeners out there, keep asking questions, stay curious, and engage in those important conversations. The future of regenerative medicine, it's being shaped right now, and we all have a role to play.
Well, it's been a pleasure joining you on this deep dive.
The pleasure was all mine. Until next time, stay curious and keep exploring the frontiers of knowledge.